Hello, this is Kristen Bender and Casey Mafucci presenting on our Lab 5 Respiration and Vital Capacity titled Just Breathe.
The focus of this experiment is on the process and functions involved in respiration. We want to understand the exchanges of oxygen and CO2 that keep the balance of pH within the blood, as well as understand the lung capacity by observing vital capacity to see the amount of air that can be exchanged by lungs within a single breath.
The objectives of this experiment are to understand acid-base balances in the body by understanding respiration and exchanges of carbon dioxide. Another objective is to observe lung capacity through examining forced vital capacity of the class, noticing any trends and influences that may affect the final vital capacity.
The proper pH of blood is maintained through blood's carbonic acid bicarbonate buffer system. It is crucial for blood pH to remain between 7.0 and 7.8 for the body to function at a normal state. This occurs through the exchange of oxygen and carbon dioxide. In this buffer system, carbonic acid is in equilibrium with dissolved carbon dioxide, which is a product of aerobic respiration. In this experiment, the CO2 in the air will be recorded and compared to the CO2 produced by respiration of one of the members of the group. These will then be compared to CO2 produced after the person exercised, observing the changes in carbon dioxide produced at normal respirations versus increased breaths. In respiration, the lungs expand to breathe to intake different volumes of air. The different volumes of air that lungs are able to intake are influenced by different variables, including age and sex. Tidal volume refers to the amount of air that lungs are able to exchange during a normal breath. Inspiratory reserve volume refers to the total amount of air taken when a deep breath is taken, and the lungs expand to a larger volume. The expiratory reserve volume refers to the amount of air that is able to be exhaled during a forced breath. These three volumes comprise the vital lung capacity. In this experiment, vital lung capacity was measured from each member of the group to observe differences in lung capacities and compare them to those of height and sex.
Hypotheses. Our hypothesis for the respiration experiment is that there will be a greater carbon dioxide concentration in air exhaled after an individual exercises as compared to resting respiration carbon dioxide concentrations. Our hypothesis of the lung capacity is that there will be a higher vital lung capacity for a taller individual.
In the respiration portion of the lab, we determined that increased activity generates more carbon dioxide production, as cells use more energy and oxygen and must maintain blood pH. For the vital capacity portion, we observed a clear relationship between height, gender, and vital capacity levels. The height of the individual appears to be directly proportional to the capacity of the lungs. Additionally, based on reference tables, males have greater vital capacities than women.
Methodology. There are four main parts to this experiment: dissection, slide identification, respiration, and vital capacity. In dissection, we followed the manual dissection procedures and were able to observe the differences in fetal blood path, lungs, trachea, brain, and spinal cord in a fetal pig. We also identified the length of the digestive tract by removing small and large intestines, unraveling, and measuring length. In slide identification, we examined human nerve, brain, and lung slides. In respiration, we utilized a carbon dioxide sensor from the Vernier Company in conjunction with the LabQuest and Logger Pro system for this portion. We established a baseline with the room air reading by setting the carbon dioxide sensor on the lab table and collecting values, waiting for approximately 90 seconds for the numbers to stabilize. After, we recorded exhaled air using a 250 milliliter plastic bottle, exhaling deeply into the bottle 5 times, recapping and then quickly replacing with the carbon dioxide sensor, ensuring a snug fit, and began collecting data. We then recorded exercise exhaled air by going outside the lab, jogging in place and doing jumping jacks for 3 minutes, exhaled into the bottle 5 times, recapped, and quickly replaced with the carbon dioxide sensor, and collected data. Data for each section recorded was until it reached a consistent plateau or at least 500 seconds was reached. Final carbon dioxide values recorded and updated in the Google Drive spreadsheet. For final capacity, we utilized a spirometer in conjunction with the LabQuest and Logger Pro system and each member exhaled and inhaled deeply as possible into the spirometer and recorded the values on the graph. Each member attempted this twice. Results and discussion. For respiration, we recorded the following values for one member of the team. The room air was around 645 parts per million carbon dioxide, while exhaled air was approximately 15,000, and exercise air was about 13,000. This was not consistent with our hypothesis. Our individual group data did not match, but the class data and averages aligned with our hypotheses. We believe this may have been a result of human error, as carbon dioxide may have escaped the bottle when inserting the sensor. As seen in the section mean and class mean and standard deviations, these are fairly consistent, and carbon dioxide is higher in exhaled exercise air. Due to increased activity, increased respiration, more oxygen is consumed, therefore more carbon dioxide is released in order to maintain blood pH. Additional factors that may affect this include athleticism, health conditions, and environmental factors. Athleticism may influence this, as individuals may more efficiently use their lungs and be able to produce less CO2 despite the greater amounts of oxygen consumed. Health conditions such as asthma and emphysema may limit the amount of oxygen that's uptook by the system and therefore reduce the amount of carbon dioxide released. Environmental factors, including smoking or pollution, may also influence this in a negative manner.
In the vital capacity portion of the lab, member 1 is a female who is 64 inches tall. According to the standards for height and sex, the expected forced vital capacity would be 3.71 liters. The average forced vital capacity measured in the experiment for member 1 for 3 trials was 4.937 liters. This is above the standard for the specified age and sex of the individual, showing a higher forced vital capacity. Member 2 is a female who is 68 inches tall. According to the standards for height and sex, the expected forced vital capacity will be 4.155 liters. The average forced vital capacity in the experiment for member 2 was 4.967 liters. This is above the standard for specified age and sex, showing a higher forced vital capacity. This can be seen in the recorded graphs for the forced vital capacity each member obtained from Logger Pro. When we compared our group's data against the histogram, our group's average vital capacity was 4.952 liters, which is above the class average of 3.6009. This is 1.3511 liters above the class average, which is more than one standard deviation away from the mean of vital capacity for the entire class. We noted there is a correlation between height and vital capacity, which also males have a greater vital capacity than females. Additional factors include athleticism, health conditions, environmental, and body structure / genetics.
Next, we observed experimental slides of brain, lung, and nerve cells.
Final thoughts. In conclusion, we were able to comprehend exchange of CO2 and respiration, observe trends in respiration and vital capacity, and effectively learn through dissection about respiratory system structures.
Below are our references.